Hi everyone, I'm Claudia Silva, PhD student in Integrative Genomics in the Universidad Mayor of Chile. Now I present my poster titled Comparative Genomics of Commensal Clostridia Isolated from the Gut Microbiota. Clostridium is a gram-positive obligate anaerobe and sporiform bacteria with bar shape considered as commensal in the gut microbiota. Several members of Clostridium produce toxin. Clostridium baratii are known for infrequent cases of bacteremia or gastrointestinal complication. One toxin produced for some Clostridium is BONT toxin. There are 9 BONT toxin serotypes with differ in their protein targets. Serotype F is generally produced by a strain of Clostridium botulinum and some strain from Clostridium baratii. Clostridium baratii produce the serotype F7. The BONT toxin produce botulism. Botulism is characterized by a flaccid paralysis due to the blocking of neurotransmitter release, usually acetylcholine, and at the neural muscular junction. For this reason, we study the genome and relevant genomics determinant among public Clostridium baratii genomes to better understand their diversity and contribution to disease. Well, we use 12 genomes. On the genome size on average was of 3 million base pair, and we have an average of 2,918 proteins on average, and all Clostridium baratii isolated in the laboratory have similar characteristics to other genomes. First, we realized the pangenome of Clostridium baratii strain. We realized a gene prediction with prodigal, and finally the core and pangenome with Rory. We found a 47.6% of core genes and open genome in the Clostridium baratii strain. With the information of the pangenome, we realized a cluster ortholog analysis, and we found that in the accessory genes, we have more categories associated to replication, combination, and repair, the L category, that suggests the genome have a variation in their repair mechanism and adaptation to stress. After, and we found more protein associated to category B, that is defense mechanism, that suggesting strain level differences in adaptation to stress. After, we evaluate the toxins present in pathogen Clostridium and Clostridium baratii. We found that the PLC toxin are present in all Clostridium baratii genomes, and in 7 of 12 genome of Clostridium baratii produce the BONT toxin. Finally, we released an BONT analysis. We download the BONT F7 cluster from NCBI. We create a database and evaluate the presence in these strains. And finally, with the BONT sequence, we released the phylogenetic analysis with the Clostridium botulinum and Clostridium baratii strains. We found that the all Clostridium baratii produce the serotype F, and the other Clostridium botulinum produce the different serotype of BONT toxin F. Finally, we found a gap in a gap of 7 amino acids in near of the active site, but this is found only in Clostridium baratii strain, and Finally, we conclude that Clostridium baratii is an unstudied Clostridium species that could pose a potential threat, considering some isolate could produce the botulism toxins. A comparative genomic analysis of 12 genomes of Clostridium baratii indicate a diverse open pangenome and diversity in adaptation to stress process. All Clostridium baratii were equipped with phospholipase assay or PLC toxin, and 7 out of 12 genomes carried a complete BONT FC cluster. The BONT amino acid was conserved among all Clostridium baratii genomes and displayed distinct differences compared with Clostridium botulinum BONT. Finally, we conclude this analysis could be an important to understand the properties of rare Clostridium taxa of clinical importance. Thank you very much.